Welcome back to this next video, and this is actually the part 2 on the monoclonal antibodies. In the first part of this video, we discussed about the monospecificity of the monoclonal antibodies. That monoclonal antibodies, they have affinity for the same antigen or for one epitope or for one type of the cell or tissue, and actually the monospecific and the monovalent terms, they were actually overlapping. We differentiated between the monoclonal and the polyclonal antibodies, that the monoclonal antibodies, they react with a specific epitope on a given antigen, while the polyclonal antibodies, they react with various epitopes on a given antigens. We briefly discussed about the history of the monoclonal antibodies, that the concept of the magic bullet, that was actually introduced by Paul Ehrlich, and he and Eli Mechnikov, they actually received the 1908 Nobel Prize using this strategy for the effective treatment of the syphilis. And then in 1970s, the multiple myeloma or the B cell cancer, they were discovered, which were used to study the structure of the antibodies. And in 1975, Kohler and Milstein, they actually succeeded in producing the hybridomas, for which they shared the Nobel Prize for Physiology and Medicine in 1984. We discussed about the production of the hybridoma, that how this is achieved, that you are actually going to inject your desired antigen into the mouse, and the mouse is going to produce the different kind of the spleen cells, which actually are the B cells, which will be producing different kind of the antibodies. And you have to combine these spleen cells with the myeloma cell. You have to fuse them utilizing the polyethylene glycol, and you will be having different kind of the hybridomas, which you will be selecting from the other kind of the cells using the hate media. Now in this particular video, I want to focus on that if you look at the mouse is actually producing different kind of the B cells against the same antigen. And if it is producing different kind of the B cells, what that means is that you are actually fusing different B cells with the myeloma cell. So in the end, the hybridomas you are getting, that will be of different types. Like for example, if you look at this one, just for the simplicity, this is a mixture of the hybridomas on the hate media. Like for example, you have got 4 different kind of the hybridomas. You've got the hybridoma 1, the hybridoma 2, the hybridoma 3, and the hybridoma 4. What they simply mean is that here the B cell that is different from the B cell that is present in this hybridoma. This B cell is different from the hybridoma, from the B cell present in this particular hybridoma. And if the B cell, they are different from each other, they are going to produce different kind of the antibodies. We will come to that in a while. If this is an antigen, for example, this is your antigen, and you have 4 different kind of the epitopes on the surface of this antigen. This is epitope 1, this is the epitope 2, this is the epitope 3, and this one is the actually the epitope 4. So I'm using this these different kind of the diagrams just to show you different kind of the epitopes present on the surface of this antigen. If you look at this hybridoma, the B cell present in this hybridoma number 1, it is producing an antibody, and let us represent that by A. So we say that the hybridoma 1 is producing the antibody A, and this antibody have got affinity for the epitope number 1. The hybridoma number 2 is producing the antibody B, and this antibody B is actually having a stronger affinity for the epitope number 2, so it can only interact with this epitope 2 present on the surface of this antigen. The hybridoma number 3 is producing antibody C, and it is interacting with the epitope number 3. Hybridoma number 4 is producing antibody D, and that is actually interacting with the epitope number 4. So you have got 4 different hybridomas, 4 different kind of the antibodies, and they are interacting with different epitopes present on the surface of this particular antigen. Now the question is how you are actually going to separate these hybridomas from each other, because initially they are present on the hate media, in the form of the mixture, so you will not be able to isolate the antibody A or the antibody B, C or D in a purified form. So what you have to do is that you have to separate these hybridomas from each other now. Now to achieve this, what you do is that you are going to dilute this mixture of the hate media. You are going to dilute this to an extent so that you are sure that in one well of a microtiter plate there is only one kind of the hybridoma. For example, these are the wells of the microtiter plate. I'm only showing you 4 wells in a microtiter plate. So what you have to do is that you have to dilute this mixture to an extent so that you are sure that in this particular well only hybridoma number 1 is present. In this particular well only hybridoma number 2 is present. Similarly, hybridoma number 3 and hybridoma number 4, all of the hybridomas now they are present in different wells of the microtiter plate. What this means is that when you have separated the hybridomas from each other, so in this particular well you have got the hybridoma number 1, so in this particular well you can expect only the antibody A, because the hybridoma number 1 is producing antibody A. So in this particular well, as only hybridoma number 1 is present, so only antibody A will be present. If you talk about this one, only hybridoma number 2 is present, so you will be expecting only antibody B in this particular well. So as the hybridoma, same will be the case for the hybridoma number 3 and the hybridoma number 4. So one hybridoma, that means only a single kind of the antibody that will be present in that particular well. The next thing is that you have to screen these antibodies. And for screening, again, one of the most famous technique that you used for the screening of the different clones for their ability to bind to the antigen is ELISA, or the antigen microarray assay. I'm not going into the detail of the ELISA or the antigen microarray assay, as our main focus here in this particular video is on the monoclonal antibodies. But one of the famous technique that you can use to screen these antibodies for their ability to bind to their respective antigen or epitope, that is ELISA, or you can use the antigen microarray assay. Now what you have to do is that you have to take an ELISA plate, and in different wells of the ELISA plate you have to introduce a specific epitope or a specific antigen, you can see. As you can see over here in the first well, you have got this antigen A. As you can see over here, this one is the epitope number 1. So in the well number 1 of the ELISA plate, you have only introduced the epitope number 1. Similarly, in the well number 2, you have the epitope number 2. In the well number 3, you have got the epitope number 3, and in the fourth well, you have got the epitope number 4. Now what you have to do is that you have to introduce the antibodies over here, and when you introduce antibodies over here, and if a color production is there when you talk about the ELISA plate, that means that that particular antibody is interacting with the epitope of interest. For example, if you look at this particular well, here you have got the epitope A, and as I've told you that the epitope A can interact with the antibody A with a stronger affinity. So when you introduce the antibody A in this particular well, this antibody is going to interact with the epitope number 1, giving you a color, which means that this antibody is actually having a very strong interaction with this antibody. And if the production of this particular antibody is high, and if the clone, by clone I mean that if the hybridoma that you have produced, if that is stable, you can actually select that for the future use. So here in the ELISA plate, you will be actually going to see that which of the antibody is having a stronger interaction with this particular antigen and with which of the epitope. So say for example, if the epitope number 1 that is having a weaker interaction with the antibody A, you cannot select this particular hybridoma for the production of the monoclonal antibodies, because if the antibody is having a weaker interaction with that particular epitope, the results that will not be reliable. Say for example, if you talk about the epitope number 2, and that is having a very strong interaction with the antibody B, that means that the antibody produced by the hybridoma number 2, you can select that for the diagnostic or for the therapeutic purposes. So what you have to do is that you have to screen each and every clone for its interaction with different epitopes, and the hybridoma that is giving you the stronger interaction with a particular epitope, that is actually used for the diagnostic or the therapeutic purposes. Now, once you have selected your desired clone, what you have to do is that the B cell that produce the desired antibodies that can be cloned to produce many identical daughter clones. This simply means that you have to increase the number of the desired hybridoma. One of the famous culture media that is used for this particular purpose, that is known as the RPMI 1614. This is a prepaid media. You can order it and you will be getting prepared media. And in this particular media, you have got the antibiotics and the fetal bovine serum. And this particular media can be used to produce the desired antibodies from the desired clone. Once you have selected your desired clone, the next step you have to do is that you have to go for the purification of the monoclonal antibodies, because you are interested in the antibodies, you are not interested in the hybridomas. So once you have selected your desired hybridoma and that produce the desired antibody, the next step you have to do is you have to purify the monoclonal antibodies which is of your interest. So the first step you have to do is that you have to remove the cell debris, the lipid or the clotting material. They are first removed typically by centrifugation, followed by filtration with a 0.45 micrometer filter. So the first step is centrifugation, followed by filtration using a particular filter. Now, as these large particles, like if you talk about the cells or the cell debris, they are very large particle. They actually cause a phenomena called as the membrane fouling, which can actually create problems in the later purification steps. So what you have to do is that you have to check your membrane each and every time. You have to check these filters whether they are fouled or not. So if they are fouled, you have actually to you have to replace them if you are using them further in the process of the purification. So once you have removed the cell debris, the next step you have to do is that the concentration of a product in the sample may not be sufficient, especially in cases where the desired antibody is one produced by a low secreting cell line. Say for example, you have selected this particular clone, the hybridoma number 2. You have selected this particular clone as this is having a stronger interaction with its epitope, but this particular clone is producing the antibodies in a very small quantity. So if you are this is the desired hybridoma, because this is giving you the desired interaction, but as it is producing a very small quantity of the antibody, so what you have to do is that you have to go for the ultrafiltration and the dialysis to get the desired antibody in a purified and in a condensed form. The next thing you have to do is that you have to remove the charge impurities like the nucleic acids or the endotoxins, and they are actually removed by the ion exchange chromatography. These are different kind of the techniques. They are very separate kind of the techniques, and I'm not going into the details of the techniques in this particular time. Now, a much quicker and single step method that is used for the separation of the monoclonal antibodies, that is known as the protein A/G affinity chromatography. And in this particular affinity chromatography, if you look at the level of purity, that is generally very high. You are actually getting a purity of about greater than 80%. However, this method may be problematic for antibodies that are easily damaged because a very harsh condition. They are generally used in this particular affinity chromatography. So most of the time the techniques that are used for the purification of the monoclonal antibodies, that is the centrifugation followed by the filtration, and then the ultrafiltration or dialysis in case of the antibodies which are produced in a very low quantity. So we will discuss this. We will continue the discussion in the next video, and in the next video I'll be telling you that what kind of the monoclonal antibodies that are available for the diagnostic and the therapeutic purposes, and what are the different types of the antibodies that you can actually use for the diagnostic or the therapeutic purposes.